Hello everyone and welcome to this video. In this particular video, we are going to talk about the affinity chromatography concept. So before going into the detail of the affinity chromatography, first we need to understand the term affinity. So affinity is the kind of a attraction. So in the context of biology, it is the attraction or the interaction between two molecules. So the affinity chromatography works on the principle of mutual recognition force between a ligand and a receptor. So the ligand is generally present on the biomolecule, or the biomolecule itself is a ligand, and the receptor is generally present on the matrix. But it's not a hard and fast rule that the ligand will be only present on the biomolecule and the receptor will be only in the matrix. It can be vice versa too. Also, the affinity chromatography works on the hydrogen bonding, van der Waals interaction, electrostatic interaction, shape complementarity, and interaction between the ligand receptor pair. So, if we look at the simple example for the receptor ligand pair, so we are having a cell, and cell has a certain kind of cell surface receptor which can accept certain kind of chemicals or the ligand, which are actually important for their intercellular signaling. So, keeping aside the intercellular signaling, if we only look at the receptor part and the ligand part, so cell having this particular kind of receptor which can only accept the oval kind of a ligand. So here we are talking about the shape complementarity. So when this ligand and the receptor will come in the close proximity, there will be interaction formed between them and they will form a receptor ligand complex. So this is the phenomena which we use in the affinity chromatography. So there is a mutual recognition force between the receptor and ligand, due to which they form the receptor ligand complex. So if we look at the kinetics, so it is the simple, as I said, receptor plus ligand forming the receptor ligand complex, and it is actually forming due to the mutual interaction between these groups, and there is associated a dissociation constant. So if these two molecules can actually bind, so they can actually dissociate too, and this dissociation is actually called the dissociation constant, which is given by the concentration of R into concentration of the L by the concentration of the receptor ligand complex. Here R is the receptor and L is the ligand. So now coming to the concept, so as I said, the affinity column has a receptor present on the matrix. So this is a column and here is the matrix, and these green beads are representing the matrix, and the oval black structure here is the receptor, and the biomolecule that we want to separate must contain the suitable ligand. So this is our protein with the suitable ligand attached to it. So how this suitable ligand is attached to it is actually through the genetic engineer, through the recombinant DNA technology, which will be talked in the upcoming genetic engineering lectures. But just for now, it's said that it is a fusion of the two proteins due to which this particular protein is made. And now this protein is with ligand, and it can actually bind with this particular receptor. So this particular protein with ligand is actually can form the affinity with this receptor. So now if we are having a mixture of biomolecules and we want to separate our biomolecule of interest, so in this case the purple one with the red ligand conjugated with it is our molecule of interest, and rest other are the uh, protein that we don't want. So how do we separate? So we are having the affinity column, and now we will run this particular protein through this column. So only the interaction between the suitable ligand receptor will take place, and there will be interaction formed between it, and the rest will be eluted out. So in the next step, what we can do is we can take another kind of ligand which will compete with the this our protein molecule with the ligand conjugated to it. It will actually compete and they will displace this molecule, and thus how we will get our molecule of interest. So depending on the interaction between the receptor and the ligand, there is actually different type of affinity chromatography associated with it. So the first one is the bioaffinity chromatography, and the second one is the pseudo affinity chromatography. So in this lecture, we are going to take a look only into the bioaffinity chromatography, and the pseudo affinity chromatography will be covered on the next lecture. So the bioaffinity chromatography actually works on the biomolecules which are used as the affinity molecules. So here in the bioaffinity chromatography, we are actually exploiting the biological affinity phenomena such as antigen antibody, enzyme substrate, or enzyme inhibitor, etc. So we are actually exploiting the antigen antibody interaction kind of a thing. So there are two classical examples. One is the GST glutathione and the another one is the nickel anti-affinity chromatography. So we are going to take a look into both of it. So first coming into the glutathione GST based affinity chromatography. So the glutathione S transferase, which is GST, is an enzyme which uses the glutathione as the substrate. So the recombinant protein which we are going to make must contain the GST tag, and the matrix is glutathione coupled matrix. So like I said, so here is the R protein and the red one here is the GST tag, and the matrix is the matrix will contain the glutathione. And this, then what we do is the same thing. The sample is loaded on the column. The affinity between receptor and ligand will take place, and the unbound protein will be washed off. Then protein will be eluted with a different concentration of the glutathione. So for example, so here if this is a R molecule and it contains the GST tag, and these are the glutathione coupled matrix. Now if we run the this molecules, so these there will be a same interaction will take place. And now what we can do is we can use the glutathione a different concentration of glutathione to elute these molecules. So that's how actually the elution works. And we can actually cut the GST tag through the thrombin. So now I'm having a question for you. So we are having a two protein fused with it, which is called the recombinant protein. So now if we cut the GST tag and now we want to elute our molecule, which doesn't contain the GST tag, how will you do that? And now actually I'm coming to the nickel anti based affinity chromatography. But for the previous question, you can comment in the comment box, or I will tell you at the end of this lecture. So the nickel NDA based affinity chromatography, here the recombinant protein must contain the histidine tag, so which has affinity towards the nickel. So the matrix here contains the nickel, and the recombinant protein that we are going to use contain the histidine tag. And the matrix is actually first charged with nickel, and then it is being used. So the receptor ligand interaction will take place between the histidine and nickel, and thus our molecule will get attached to the matrix. And now again to elute the molecule we use the another reagent. So the this reagent is the imidazole. So the imidazole actually compete with the histidine to bind with the nickel, and thus we get our molecule eluted out. So I hope you understood the affinity chromatography. Now coming to the question based on the glutathione GST based affinity chromatography. So now we are having a two protein fused together. One are normal protein and another is the tag, which is the GST tag. Now if we use the thrombin to cut it, so the GST tag will be removed. So now we can again use the glutathione GST based affinity chromatography. So what will happen is only the GST tag will be bound to the matrix and rest of the protein will get eluted out. So thus we will get our original molecule of interest without the GST tag on it. So I hope you liked the video and you understood the video. If you did, please like, share and subscribe, and I thank you for attention. And if you have any question, you can put in the comment box. And for the notes, you can follow us on the Instagram. The Instagram ID is the Crispier Biotech. And thank you.